Hello, my name is Joshua Mugabe. I welcome you to Coordination and Control, questions 11 to 15. Question 11. Describe the structure of the rod cell of the mammalian eye. Part B. How are the rod cells modified to perform their function? Describe the structure of the rod cells of the mammalian eye. We have the rod cells. They are cylindrical structures. So let us look at them here. We have two types of cells. We have the rod cells. Rod cells, these ones that we have this side. These are the rod cells. We shall leave the one on the sides. This one we shall leave it out. This one is a cone cell, but we need to concentrate on the rod cell. Now, when you look at the rod cell, you can see that the rod cells, they are cylindrical. You look at their shape, they are cylindrical. The outer segment of the rod cell is long, slender, and rod-shaped, and is in contact with the pigmented epithelial cells. So when you look at the outer segment is this part that we are seeing here, from here to here. That is what we call the outer segment. All this part here, all that one is what we call the outer segment. And we are saying that the outer segment of the rod cell is long. When you look at it, you see that it is long and it is slender. It is slender in size and rod-shaped. It is rod-shaped. And we say that is in close contact with the pigmented epithelial cell. So here we have our pigmented epithelial cell, which is the colloid layer. Here it is where it is attached. The outer layer, that's where it is attached. The outer segment has membranous discs that contain rhodopsin. They are discs that we are seeing that are moving across. Those layers that we see that move across, those are the discs. And those discs, they contain rhodopsin. Then we have the inner segment is connected to the outer segment by means of modified cilium. So when we come to the inner segment now is this part here, which we have here as the inner segment. From here, come like that. That part there is the inner segment. And it is connected, we are saying that it is connected to the outer segment by means of modified epithelium. So where are those cilium? Cilium are these black lines that we see. We say that this one all of this one is the inner segment, and this one up is the outer segment. So the inner segment is connected to the outer segment by modified cilia. Modified cilium, they are these ones that we are seeing, the black structures coming from the inner segment going to the outer segment. The inner segment contains many organelles with large number of mitochondria. So we are looking at the inner segment. Our inner segment, we are saying, is this one here, and it contains many organelles, which includes the mitochondria. These are the mitochondria. Stop. A slender fiber called the rod fiber arises from the inner segment of the rod cell and passes to the outer nuclear layer through the external limiting membrane. In the outer nuclear layer, the enlarged portion of this fiber forms the cell body or rod granule, which contains the nucleus. So we have, this is our nucleus, where we are having, coming from the inner body, where we have our nucleus, the cell body. The synaptic terminal, thick fiber, arises from the cell body, passes to the outer plexiform layer, and end in a small and enlarged synaptic terminal or body. So from here, from our cell body here, where we have our nucleus, we end up going to an enlarged portion. The enlarged portion is this part here. That enlarged portion is what we call the synaptic terminal. These ones are synaptic terminals. Synaptic terminal of the rod synapse with the dendrite of the bipolar cells and horizontal cells. So this synapse, which we are seeing around here, all these ones, we have the synapse, these are the synapse, and they have got dendrites in them. Those dendrites, they go to the bipolar cell. This one is the bipolar cell. You can see it has got the dendrites. Those dendrites of the bipolar cell, they go into the synaptic terminals. And also, they are what we are calling the horizontal cell. The horizontal cell is this one. It also has dendrites which go into the synapse of the synaptic terminals. The synaptic vesicles present in the synaptic terminal containing a neurotransmitter glutamate. So these synapses contain synaptic vesicles, which synaptic vesicles contain a neurotransmitter substance which is called glutamate. Part B. How are rod cells modified to perform their function? The rod cells do retinal convergence, in which several rod cells synapse with a single bipolar neuron. So here what they are saying, we have here several rod cells. All these rod cells that you are seeing here, these 3 rod cells, they end up synapsing with one bipolar body. So this one will come and form a terminal synapse here. And also this one forms a terminal synapse, and then they are connected to dendrites. But those dendrites, they join together and they join to one bipolar neuron. So that's what we are saying, that the rod cells do retinal convergence. That one whereby they all connect to one bipolar neuron is what we call retinal convergence. In several rod cell synapse with a single bipolar neuron. This arrangement increases the eye's sensitivity to light. On exposure to low intense light, each rod cell generates its own receptor or generator potential. So here we have, like for example, here we have 3 rods. Each one is going to generate its receptor or generator potential. And all of them, when they generate, all of them, their potential will be joined together and passed over to one bipolar neuron. So it will be like summing up so that they can increase the action potential. Sub-threshold in magnitude, these spatial summate form one large receptor potential, whose magnitude is either equal or beyond the threshold establishes an action potential that fires an impulse across the adjoining bipolar neuron. So here, when the 3 of them fire, when they do fire or they do generate the generator potential, each one contributes its generator potential. And that is what we are calling the spatial summation. And all of them, they will go to one bipolar neuron. Now, when they combine the generator potential, the generator potential will either be equal or beyond the threshold needed to establish an action potential that will fire the impulse across the bipolar neuron. And therefore, the information will be sent to the brain via the optic nerve. So when the impulse is generated through here, through summation, through the bipolar neuron, that information is carried to the brain via the optic nerve. Question 12. Describe the structure of the mammalian retina. Part B. Explain how fast speed of impulse transmission is ensured in the nerve cells. And part C. Describe postsynaptic changes occurring in excitatory and inhibitory synapse. Inhibitory. Describe the structure of the mammalian retina. The retina consists of the photoreceptor, or what we call the outer layer. This one having the cones and the rod, which are partly embedded in a pigmented epithelium of the choroid. So when we look at our eye here, we are going to get this eye, and we are going to cut a small piece. When we cut this smaller piece, it is the one which we are going to enlarge to give us this whole part that we are seeing here, which is going to be representing our retina. And we are saying the retina consists of what we call the photoreceptor, or the outer layer. Here we have what we are calling the outer layer. Here, outer layer, and this outer layer is the one that has got what we call the photoreceptors. And the photoreceptors, we have the cones, these ones here, with a cylindrical, those are cones, and then it is also made of the cones are these ones that are cone-shaped, these ones that we see as cone-shaped, they are the cones, and the rods are these ones that are cylindrical. So they are in a region which we call the outer region of the retina. It is the one which is containing these photoreceptors, the cones and the rods. That we say that next is the intermediate, or the middle layer, and this one contains the bipolar neurons synapsing with the photoreceptor cells. This layer contains also the horizontal cells and the macrine cells. So we have now the middle layer. The middle layer comes from here and it goes up to here. That is our middle layer, and it has got what we are calling the bipolar cells. These ones we are seeing in the middle here, all these ones are the bipolar cells, and it is also made up of the macrine cells. The macrine cells are the ones that we are seeing somewhere here. Those are the macrine cells. They are found in what we call the middle layer of the retina. The innermost layer is the ganglion layer, and this one contains the ganglion cells with the dendrites in contact with the bipolar neurons and with the axon of the optical nerve fiber. So we have the innermost layer. Innermost layer is this one here. And this innermost layer, we are saying it contains the ganglions. These are our ganglion cells, which have got dendrites. These dendrites which are connecting with, or they are in contact with, the bipolar cells, and they also have the axons, these axons of the optic nerves that are taking the message to the brain. That is what we call the innermost layer. Explain how fast of the impulse transmission is ensured in the nerve cell. There is what we call myelination. Myelin sheath covers the axon of the nerve fibers and is only absent at node of Ravian. The myelin sheath stimulates the axon. Ionic exchanges, depolarization, and repolarization occur only at the node of Ravian. Now let us look at our neuron here. When you look at this neuron, we have this part here that we are seeing here. That part is an axon covered by a myelin sheath. That outer layer is what we call a myelin sheath. Now when you cut this neuron through like this, and then from here, this is what you are going to see with the part that has been cut. This is what you're going to see. Now when you look at it in the middle of the neuron, that is what we call the axon. And then the one part which is covering the axon, that is what we call the myelin sheath. So this is what we are calling the myelin sheath that covers the axon of the nerve fiber, and it is absent at the point we call the node of Ravian. Now at this point you can see there is no myelin sheath here. That is what we call the node of Ravian. When you look here, there is no covering here. The axon is not covered at those points. Where the axon is not covered, that is what we call the node of Ravian. So this myelin sheath here that we are seeing, it insulates the axon. It covers the axon, and it also allows ionic exchange. Ion exchange, sodium, potassium in between the node of Ravian, depolarization, repolarization, all of them, they occur at the node of Ravian. So we shall see that it is going to do a very good work by helping the depolarization to take place very fast. So the impulse will be just jumping from one node of Ravian to another node of Ravian instead of passing through the axon direct. Action potential, therefore, just leaps from one node of Ravian to another, and the speed of the impulse transmission increases tremendously because of the saltatory transmission. So the impulse, when it's generated from this side here, when it reaches here, the impulse, when it comes from wherever it is generated from, when it is coming through, it will not pass through directly inside the axon because that one is going to delay. So it is going to just jump from one node of radian to another node of radian like that. It will just keep on jumping from the node of radian, and therefore we shall see that the impulse speed will be tremendous because of the saltatory transmission, that jumping from one node of radian to another node of radian is what we call saltatory transmission of the impulse. It increases the speed of transmission. Increasing the axonal diameter. So one of the ways of trying to increase the speed is to increase the diameter of the axon. The axon layer there, if we increase the diameter, then the speed is going to be very fast. The larger the axon, the faster the speed of impulse transmission. Giant axons offer minimal longitudinal resistance to the flowing impulse within the axoplasm. So if we have the axon diameter increase, the more you increase the axon diameter, the less resistance is received by the impulse which is passing through. So when the impulse is passing through in a very giant axon, it receives less resistance from the horizontal flow of the axoplasm. The length of the membrane affected by the lock circuit increases, thereby increasing distance between adjacent depolarization. Another one is increasing the temperature. Increasing the temperature increases the diffusion rate of ions across the axomembrane and the rate of the enzyme-controlled reactions. This speeds up the depolarization and repolarization, establishes a local circuit fast enough, thereby increasing the speed of impulse transmission. So we are saying here that if we increase the temperature, by increasing the temperature, we are going to increase the rate of diffusion of ions across the axon membrane. And we know that most of the enzyme-controlled reactions, as the temperature increases, the rate of enzyme activity increases. So this one is going to speed up the depolarization and repolarization to establish local circuit fast enough so that there is increase of the impulse transmission. So one of the ways of speeding up the impulse transmission is to increase the temperature. Part C. Describe postsynaptic changes occurring in excitatory and inhibitory synapse. At the excitatory synapse, reception of the neurotransmitter substances, that is acetylcholine, on the receptor side, changes their configuration such that the membrane channels in them are opened up, therefore allowing sodium ions to diffuse through the postsynaptic membrane into the postsynaptic crypt or synaptic knob, and making it more positive. So here we are seeing at the excitatory synapse, we are having our synapse across here, and when the reception of the neurotransmitter substance are released, they are going to be received by the receptor on the postsynaptic side. And this one will cause the change the configuration of the membrane, and then it will cause the membrane channels to open and allowing in the sodium ions to enter into the postsynaptic membrane. And when this one happens, it is going to cause this part to become more positive. If a threshold value is reached, potential difference of the membrane therefore changes, and an excitatory postsynaptic potential EPSP results. This fills up the threshold is reached, which results into the action potential being fired in the postsynaptic neuron. At that point, the impulse has crossed the synapse. So, due to the entry of the sodium ions entering into the postsynaptic knob, time is going to come when we get the threshold. And when we reach the threshold, we are going now to create an action potential in the postsynaptic neuron, and that one will fire an impulse which will move across the axon. Let's now look at the inhibitory synapse. At the uninhibitory synapse, release of the transmitter substance, which is noradrenaline or glycine or GABA, into the synaptic cleft leads to the opening up of the chloride ion channels in the postsynaptic membrane, resulting into chloride ions entering and potassium ions leaving. So at the inhibitory synapse, here is where we are having our inhibitory synapse. When the neurotransmitter or transmitter substance is released, and that transmitter substance which is released is noradrenaline. This noradrenaline is going to cause the postsynaptic membrane to open up the chloride ion channels. And when the chloride ion channels open, the chloride ions are going to enter. These chloride ions will enter into the postsynaptic membrane. And when they enter inside, because the channels are open, the potassium ions are going to move out of the postsynaptic membrane. They will move out of the postsynaptic membrane, and as they are leaving out, they are going to cause an inhibitory state. As a result, the interior of the postsynaptic membrane becomes more negative, and this increases the threshold, making the postsynaptic membrane harder to be excited. An inhibitory postsynaptic potential has been established. We have seen that we have allowed the potassium ions to enter, chloride ion solid to enter, and when the chloride ion enters inside, the postsynaptic membrane becomes more positive. And in addition to that, also allowing the potassium ions to go out that one is also going to make the postsynaptic membrane to become more negative. And when it becomes more negative, therefore, we have created an inhibitory postsynaptic potential. IPSP has been established, so no impulse is going to be passing that area. Question 13. What is meant by an organ of Corti? Part B. Describe how the low-pitched, loud sound is perceived by the human ear. Part C. Explain why someone who has been constantly rotating feels dizzy when he suddenly stops. What is meant by an organ of Corti? The organ of Corti is a unit in the cochlea of the inner ear, consisting of the basilar membrane, the sensory hair cells, and the tectorial membrane, where transduction of sound waves into electrical impulses occur. So when we look here, the whole of this one is our organ of Corti. And we are saying that the organ of Corti is a unit of the cochlea, and it is made up of what we call the basilar membrane. The basilar membrane is this membrane that you are seeing down here. All that membrane is what we call the basilar membrane. And then it also has the sensory hairs. The sensory hairs are these ones that you are seeing in the middle. All those ones here are what we refer to as the sensory hairs. And it is made up of the tectorial membrane. The tectorial membrane is this upper layer that you are seeing here. That is the tectorial membrane. And in this organ of Corti, the organ of Corti, it is where there is transduction of sound waves, the waves that are in the endosphere are going to be converted into electrical impulses, which will be taken to the brain for interpretation. Part B. Describe how low-pitched, loud sound is perceived by the human ear. The sound waves get collected and concentrated by the pinnae and forwarded to the external auditory meters and causes the vibration of the tympanic membrane, which also transmits the vibration to the ossicles for independence matching, that is amplification by 22 times. So when the sound is coming, will come from the outer part of the ear, which we call the pinna. So that sound, when it comes, it passes through the auditory canal, through the external auditory canal, it will find what we call the tympanic membrane, the ear membrane. It will cause it to vibrate, and it will transmit the vibration to the ossicles, that is the malleus, the incus, and then the stapes. This one is going to amplify the sound by 22 times. The amplified sound waves are then forwarded to the oval window, which produces the vibration in the pelleaf of the vestibular canal and transfer the pressure waves via the resina membrane to the endoleaf of the middle canal and then to the pelleaf of the tympanic canal. The vibration are then finally forwarded to the basilar membrane. So, when the amplified sound is forwarded to the oval window, the oval window is going to produce a vibration in the pelleaf. It will produce the vibration in the pelleaf of the vestibular canal. Then this vibration will be passed over to the middle canal into another fluid which we are calling the endoleaf. So in the middle canal, the vibration of the fluid or the endoleaf will then again be passed over to the tympanic canal, which has got also a fluid called the pelleaf fluid. And this vibration now will be passed over to the basilar membrane, our basilar membrane. So the basilar membrane is this layer here. This layer here is our basilar membrane. So it will be passed over to this membrane here, the basilar membrane. Vibration of the basilar membrane induced by the pressure wave pushes many sensory hair cells near the apex of the cochlea helicotrema, causing them to shear against the tectorial membrane. So we have seen that the vibration was coming from the vestibular. It is passed on to the middle canal. From the middle canal, it is passed into the tympanic canal. Then the tympanic canal passes it to the vestibular. Now, when it is passed over the basilar membrane, on the basilar membrane, there are very many sensory hair cells. These ones that we are seeing here, they are the sensory hair cells, which are squeezing or shearing against the tectorial membrane. The tectorial membrane is above. The sensory hair get depolarized. A generator potential are created. It builds up a threshold and initiates an action potential that fires an impulse across the axonal membrane to the cochlear nerves, vestibular cochlear nerve, to the brain. A loud high-pitched sound is interpreted by the brain. Now, because of this vibration, which goes to the basilar membrane, And we know that in the basilar membrane, we have very many sensory cells with sensory hairs. Now, when it vibrates, it causes them to shear or to push them into the tectorial membrane. And when these sensory hairs are pushed into the tectorial membrane, they get depolarized and a generator potential is created. When this generator potential builds up to a threshold, it initiates the action potential. And this action potential now will fire the impulse across the axonal membrane, and it will pass through the vestibular cochlear nerve to be taken to the brain for interpretation. The basilar membrane becomes broader and more flexible as it passes from the base of the cochlea to its apex, helical tremor. So the basilar, this one comes, when it comes from the base, it becomes wider when it is coming this side to the apex. You can see it becomes wider when it is coming to the apex from the base of the cochlea. And the sensitivity of the vibration of different frequencies change along the length. So along all this length, from this side, the base, to the exterior or the apex, we can see that it has got sensory cells and sensory hair. So here, the sensitivity keeps on varying as you move from the base going to the apex. High frequency or pitched sound are interpreted if the hair cells at the base of the cochlea are stimulated. So if most of the base hair at the base here of the cochlea are stimulated, then we are going to get a high frequency sound if these cells are the ones that are more stimulated at the base. Low-pitched sound are interpreted by hair cells at the apex of the cochlea are stimulated. So if we have those ones at the apex, if these ones at the apex are the ones that are more stimulated, then we are going to get a low-pitched sound. And the middle frequency sound are going to be interpreted by hair cells between the apex and the base being stimulated. So if we have the base here and we have the apex, then now in the middle we have the middle hair. If we stimulate these ones in the middle more, then we are going to get a mid-frequency sound. Intense loudness depends on the number of hair cells stimulated. So the more number of cells stimulated, the more loudness. Fewer hair cells stimulated, low intense sound is interpreted. If many cells are stimulated, they generate several receptor potentials, spatial summit, and a loud high intense sound is detected. Part C. Explain why someone who has been constantly rotating feels dizzy when he suddenly stops. When the head is constantly rotated, the endoleaf in the semicircular canal moves with a constant velocity, similar to that one of the head. The fluid moves with zero acceleration, and the brain interprets static equilibrium. So here we have the pupil. The pupil is in the ampulla, and in the ampulla it is filled with the endoleaf, the fluid. So when the head keeps on rotating, also the endoleaf will rotate in that direction, moving at the same speed as the head. Now the brain will interpret it as if it is stationary, as if nothing is moving, because everything is moving with the head in the same direction and at the same speed. When the rotation stops suddenly, inertia exerted on the endoleaf makes it to continue with motion, and the pattern of the impulses are taken to the brain, which interprets dynamic equilibrium when the person is resting. Now, if we have been having our head moving around, and the endoleaf is also moving around, when the rotation of the head stops suddenly, that one is going now to cause the endoleaf to continue rotating in that direction. Yet the head has stopped. And because the head has stopped, the head is stationary, but the endoleaf is still continuing in that direction, then the brain is going to interpret a dynamic equilibrium when someone is resting only. But here it will note that there is change in what is happening at the sensory cell. This accounts for the temporary dizziness. Question 14. Describe the characteristics of receptor cells. Part B. Describe the role played by the following in the maintenance of balance of the human body. Number 1, semicircular canal. Number 2, utricus and circulus. Describe the characteristics of receptor cells. They are specialized in structure and function. They possess a resting potential when they are not stimulated. They transform energy in the stimulus into an action potential when the receptor is stimulated. Each receptor produces a generator potential when stimulated. Each receptor has a threshold value of stimulation. They undergo adaptation, that is, they fail to respond to repeated stimulation. They obey all or nothing law. They are sensitive to low intensity stimulation. They have a general structure features, but one end is frequently extended, which is the oxygen. Part B. Describe the role played by each of the following in the maintenance of balance in a human body. We shall start with Roman number 1, the semicircular canal. The end of each semicircular canal has sensory cells. These are embedded in the pupil. So let us look at the end of this semicircular canal. And this semicircular canal has got what we call the sensory cells. These are our sensory cells, which are embedded in the pupil. The whole of this one is called the pupil. Movement of the head in a particular direction activates one of the canals. The pupil gets deflected in the direction opposite to the one of the head movement due to inertia in the endoleaf in the canal. So let's suppose here our head is moving in that direction. If the head is moving in that direction, what is going to happen, because the whole of this area here is what we are calling the endoleaf. The endoleaf is filled with a fluid, and that fluid we are calling it the endoleaf. It is in the ampulla. Now what is going to happen is that if the head is moving in the other direction, also the fluid will move in that direction, but the pupil is going to move in the opposite direction. Our pupil now, for it will move in that direction. As the head is moving in the other direction, the pupil is moving in that direction due to inertia. Consequently, sensory hairs are stretched. They create a receptor potential, which on building up to a threshold creates an action potential that fires impulse to the vestibular nerve fibers of the vestibular cochlear nerve to the brain, which will interpret the movement of the head. So we'll say that our head is moving in that direction. When it moves in that direction, the pupil is going to move in this direction because of the endoleaf. And our now sensory hairs are going to be stretched. When they become stretched, they are going to create a receptor potential. When they create a receptor potential, and if that receptor potentials build up to a threshold, it will create the action potential. And this action potential will now be carried by the vestibular nerve and taken to the vestibular cochlear nerve to be taken to the brain for interpretation on the movement of the head. Now let us look at the atricus and circulus. Atricus and circulus, these ones detect the position of the head, and they consist sensory hairs embedded in the otolith, calcium carbonate, which responds to the pull of gravity. So let us first of all try to understand here. We have up the atricus here and also here. So here we have this part here is what we call the otolith, and is made up of calcium carbonate. Then we have these lines here. Those are our hair processes. And this one is our sensory cell. So when the head is moving, the sensory hair cells embedded in the otolith, and this one responds to the pull of gravity. When the head is stationary, static, in the upward position, the otolith set balance over the sensory hair under the influence of gravity. So when you look here, if the head is upright, the otolith is sitting or is balancing on top of the sensory hair, and it is pressing on the sensory hairs under the influence of gravity. And that one will record the position by the sensory cells, which will be transported to the brain, and it will interpret the position of the head. The atricle responds to vertical movement of the head, such that when the body is upside down, the otolith produces a stimulation when its receptors hair are pulled downwards. Now, when we come here and we see that the head now is upside down, we can see that the otolith is now down, meaning that it is pulling the receptor hairs downwards. And that one now is going to cause a stimulation which is going to be different from the one it produced when the head was upright. And that stimulation will be interpreted by the brain in a different way. The sacral responds to the lateral or sideways movements of the head, that is tilting of the head distorts the sensory hairs. Impulses are produced, which passes to the auditory nerve of the brain, where orientation and position of the head is perceived. So when we look at the saccules, for them, you can see how they are arranged, and for them, they record the lateral movements, changing in different direction laterally. So they will record the lateral movements or the tilting of the head. And when the head tilts, it is going to distort the sensory hair. So the impulse will be fired and it will be passed to the auditory nerves, to the brain, and the orientation, the position of the head will be interpreted. Question 15. Explain the functioning of the autonomic nervous system. Part B. What are the nervous system changes that occur when the individual suddenly touches on a sharp object? Part C. Outline the differences between gray matter and white matter. Explain the functioning of the autonomic nervous system. The autonomic nervous system, or what we call ANS, forms part of the peripheral nervous system, and that controls involuntary activities of internal organs and smooth muscles. There is no involvement of the higher brain center, what we call the cerebral cortex, except for the smooth muscles of the anal and the urinary sphincter, which are partly under voluntary control. So we have the autonomic nervous system, and this one is part of the peripheral nervous system, but it controls involuntary activities of internal organs and some smooth muscles, where there is no involvement of the higher brain center, or what we call the cerebral cortex, apart from some few smooth muscles of the anal and the urinary sphincters that are also partly under the voluntary control. The autonomic nervous system is made up of the sympathetic nervous system, SNS, and the parasympathetic nervous system, PNS. And each visceral organ receives nerves from both. Preganglionic fiber of both are cholinergic. They release acetylcholine. So what do we call preganglionic? Let's try to look here. Here we have the parasympathetic, and here we have the sympathetic. This is a ganglion of the parasympathetic, and this is a ganglion of the sympathetic. Now I want you to see the movement from the central nervous system to the ganglion, the distance. That distance from the central nervous system to the ganglion is what we call the preganglion. Here, preganglion. So you can see the preganglion of the sympathetic is short, and preganglion of the parasympathetic is long. Now, when this neuron reaches into the ganglion, there is going to be formation of a synapse going to another neuron, going to the effector, even here in the ganglion. And synaptic cleft there is a release of a transmitter substance. And that transmitter substance released there in both of them is the acetylcholine. Sympathetic nervous system fiber rises from the thorax and the lumbar section of the spinal cord. So the parasympathetic, the sympathetic nervous system fibers, they come from the thorax and the lumbar section of the spinal cord. Noradrenergic, that is, they release noradrenaline at the effector organs. So for parasympathetic, when they move from the ganglion, they go to the post part here, postganglion. This neuron at this end, it releases a transmitter substance which we call noradrenaline. That is the SNS, sympathetic nervous system. And that noradrenaline is generally stimulatory or acceleratory, meaning that it increases the heart rate, increases the blood pressure, increases the ventilation rate, it causes pupillary dilation and ejaculation, and many others. However, the gastrointestinal tract, GIT, CNS decreases GIT secretion and motility and movement. So you can see that sympathetic is more of stimulatory or acceleratory. It increases the activities of the organs, accelerates, it stimulates the activity of the organs, apart from some few like the gastrointestinal tract, where it decreases its activity of secretion of enzymes. SNS, sympathetic nervous system, dominates during activities. So it is more dominant during activities. Parasympathetic nervous system constitutes of the cranial nerves, those from the brain. Most of its nerves are coming from the brain, and it is cholinergic postganglionically at the effector organ. So at the effector organ, this side, the parasympathetic nervous system produces a transmitter substance which is acetylcholine, and generally it is uninhibitory, except for the gastrointestinal tract, where it increases the gastrointestinal secretion and the motility. Parasympathetic nervous system decreases the heart rate, it decreases the blood pressure, it causes the ventilation rate to be low, it causes pupillary constriction and erection. Parasympathetic dominates at rest. What are the nervous system changes that occur when the individual suddenly touches a sharp object? A spinal reflex occurs. Pain receptors in the hand will be stimulated. They will send impulses to the sensory nerve fiber running through the dorsal horn of the spinal cord, and the impulse gets relayed to the motor nerve fiber by the interneuron or intermediate neuron that traverse the spinal gray matter. So here we have our stimulus here, the pin is going to act as a stimulus, and it is touching the finger. In the finger we have the sensory nerves. Those sensory nerves are going to be stimulated and they will create an impulse which is going to be transmitted through the sensory neuron, and it will pass through the dorsal part of the dorsal root of the dorsal root of the spinal cord, pass through the white matter to the gray matter, and at the gray matter it is going to pass over the impulse to the relay neuron, which we call the interneuron or intermediate neuron, which will also relay or pass over the impulse to the motor neuron, and the motor neuron will pass through the ventral root of the spinal cord, and it will take the information to the muscle, which are the effector. Now here it is going to cause the biceps to contract and the triceps to relax, which is going to cause the hand to be pulled away from the pin, which is the stimulus. The motor fiber running in the ventral horn of the spinal cord stimulates the skeletal muscle of the effector organ or the foot or the finger, initiating a rapid sudden withdrawal of the foot. The ascending fibers from the spinal white matter carry the sensory information to the higher center in the brain, which is interpreted. Then it will be carried back by the motor descending nerve fibers to the spinal cord, and finally to the region of pain. Pain is thus felt a few seconds after a reflex. So a reflex is this one that we are having here, moving very fast and then coming back and then pulling away the hand. That is a reflex. But when that impulse comes here, before it passed here, some of it will be taken to the brain for interpretation. And when it is taken to the brain, it will be interpreted. It will be brought back very fast, and then it will be sent to the point of pain. So what is going to happen first is that immediately someone touches the pin, he will remove because of the reflex arc. Then after immediately removing, then you will feel the pain. And that feeling of the pain is because of the impulses that went to the brain have been interpreted and they have come back to the point of stimulation. Now you feel the pain a little bit later after withdrawing the hand. Outline the differences between the gray matter and the white matter. The gray matter, now when we first of all, let's look here in our diagram here. This is our spinal cord. This is the dorsal ganglion, dorsal root. This is the ventral root. And in the dorsal root, it's where our sensory neuron passes. When it comes here we have the gray matter, this dark part where we have the gray matter. And this white part here, that's where we have the white matter. So here we are saying that the gray matter in color is due to high concentration of neuron. This gray part here, there are a lot of neurons here, the relay neurons and intermediate neuron and dendrites that are coming from sensory neuron, and then those synapses that are coming from the motor neuron. So they are ones that are causing it to become gray in color. Whereas the white matter is white due to concentration of oxygen. The white matter, this one which is white, it is mainly concentrated with a lot of oxygens of nerves going to the brain and then also other nerves going to the body. It consists of the cell body, that is the gray matter, dendrites and synapses of the brain. So the gray matter here, it has got a lot of cell bodies, like those ones of the relay neuron. It has got a lot of dendrites that are coming from sensory neuron. It has got a lot of synapses, those ones that are passing to the motor neuron, and others going to the brain. Some of them going to the brain are also in the gray matter. Whereas the white matter consists of nerve fibers arising from the nerve cells present in the gray matter. So the white matter here, it has mainly nerve fibers. Like for example, if you see this nerve fiber going to the brain and the other one coming from the brain, all those ones are in the white matter. The gray matter contains numerous intermediate neurons. So the gray matter here, it has a lot of intermediate neurons, these ones in the middle. It has very many of those neurons. Whereas the white matter consists of neurons connecting to various parts of the body and with the brain, and links the brain to the spinal cord. So the white matter, that white matter is mainly made up of oxygens or the neurons connecting to different parts of the body. Hello, my friend. Thank you very much for participating in coordination and control. Question 11 to question 15. Please do like, share, and subscribe.